It's my great pleasure to be here to present in the SGP EpIC seminar series. Uh, so I think I still remember when I was a postdoc in 2023 in Boston, attending the seminar in the lab as an audience. And now I'm presenting here as a speaker, so it's wonderful. So thanks so much for the organizing committee and the sponsors for making this possible. And today I want to share with you our published work and also some unpublished data on the structural and functional basis of mechanosensitive TMEM63 channelopathies. So in July 2023, Swesta Murthy from Volume Institute in Oregon already gave a great talk on a similar topic. So if you missed her talk, I would recommend you to go to the EpIC YouTube channel to see it. And the data I'm going to present you today will represent some of the progress that we have made since 2023. So some of you may not quite familiar with mechanotransduction. So first, I want to give you a little bit of background. Some physiological processes, such as I'm showing here, like the sense of touch, hearing, and mechanical pain sensation, you can say they obviously involve detection of physical force. And now we already have good understanding of what mechanosensitive cells and proteins are involved in touch and hearing, which I will talk about briefly in a minute. The mechanical pain is still something that we don't quite understand. But on the other hand, emerging evidence indicate that mechanotransduction is also involved in many other physiological processes, such as neural development. And this is the main focus of my lab currently. So we're trying to figure out how mechanotransduction contributes to normal neural development and when it goes wrong and how it can cause neurodevelopmental disorders. So a fundamental question is that how cells can sense force. The cells in our body can not only sense chemical clues, but also physical force in different types, such as mechanical compression, membrane stretch, bending, or shear stress induced by fluid flow. And the cells can convert this physical force into biological signals, and this is called mechanotransduction. I would say one major mechanotransduction protein is mechanosensitive ion channels. So these are the pore-forming proteins sitting in the membrane in response to the physical force. And they work actually in a very simple way. So they switch between its closed and open states. And once the pore is open, it typically allows cations such as sodium and calcium to flux into the cells. And this in neuronal cells can initiate action potentials. And in non-neural cells, it can initiate calcium signaling pathway. So in mammals, I should say there are three major families of mechanosensitive ion channels that have been identified. The piezo channels, especially piezo2, is the receptor for the sense of touch. But they are also broadly involved in proprioception, blood pressure regulation, and different interoceptions. And now this TMC channel is very specifically involved in hearing, and we don't see they are involved in other physiological processes. And this TMEM63 family are more recently identified. And now we still know very little about this family compared to piezo and TMC. But we think that these proteins may mainly involved in the neural development, which I will talk about later. And first, let me briefly introduce these piezo and TMC channels. So the piezo channels were identified by Alan Palapolian's lab at Scripps Institute in California. As I mentioned, the piezo2 is the receptor for the sense of touch. These channels are present at the nerve terminal of the somatosensory neurons underneath the skin. And normally at the touch point, these piezo channels are activated. So they convert the physical force into electrical signals that are then transmitted into our brain. And we can isolate these somatosensory neurons and put them into a Petri dish, as you can see here. So now we can use patch-clamp electrophysiology to measure activation of these mechanosensitive ion channels. So here, this is a recording pipette, and then we can use another glass probe to poke the cell membrane. So in this way, we can deliver the force to the membrane to activate those mechanosensitive ion channels. So you can say with increase in Poynting distance, we can get this larger and larger inward currents. And then how piezo channels sense force. Showing here, this is a cryo-EM structure of the piezo1, which shows that the piezo proteins they form a giant ball-shaped tremor within the membrane. So the idea is like in the resting state or the closed state, the piezo channels naturally bend the membrane from a membrane curvature. And then when the force is applied to the membrane, it will flatten the membrane and activate these piezo channels. And then the TMC channels, especially TMC1, has been established as the receptor for the hearing through, I should say, the last almost half a century efforts by many scientists, including Jeffrey Hall and Albert Fatty-Place, as I'm showing here. The TMC1 channel is expressed in the sensory hair cells in the inner ear, and they are activated by the sound-induced vibrations. And showing here is a picture of the stereocilia of Wang hair cells. And we can also isolate these hair cells and put them into a Petri dish. And we can use a glass probe to deflect its hair bundle, as you can see here. And then we can record this inward currents only when the hair bundle deflects into one direction, but not to the other direction. It turns out that the TMC protein is present at the tip of the short stereocilia, as shown here, indicated by the green dots. And they are collected to a labeling taller stereocilia through a filament called tip link. And only when the hair bundle is deflected into one direction, as shown here to the right, the tip link will generate the pulling force to activate the TMC1 channel at the tip of the short stereocilia. Okay, so now my lab here at UW Medicine, we focus on the TMEM63 proteins, which we think are mainly involved in neurodevelopment based on the clinical evidence. So you can see here that mutations in all three TMEM63 genes has been identified to be involved in neurodevelopmental disorders. Especially the TMEM63B mutation cause developmental and epileptic encephalopathies, or DEE. And the patients with this disease generally exhibit a wide range of symptoms. And these include quite a severe early onset epilepsy, intellectual disability, motor impairment, poor language skills, and dysphagia, which is a difficulty in swallowing. So this evidence, I think, clearly suggests that the TMEM63 proteins play critical roles in the brain development. And we think that by studying these TMEM63 channelopathies will give us some information on how this protein sense force and how they may function in vivo in the brain development. So today I'm going to first talk about a previous publication on the TMEM63 wild-type protein structure and function. And then I will focus on our recent publication on the TMEM63B channelopathy caused by the B44M mutation. And then last, I will talk about some of unpublished data on the TMEM63A G567S disease mutation. And in the end, I hope that I can convince you that there is a unifying structural and functional basis for the TMEM63 channelopathies. Okay, so first characterized by Swester and Adam, the TMEM63 together with OSCA was proposed to form a large family of mechanosensitive ion channels. The OSCA protein only exists in plants with 15 members identified in Arabidopsis thaliana. And Swester did a foundational work demonstrating that OSCA protein form bona fide mechanosensitive ion channels. As you can see here, but it's beautiful electrophysiology recordings. And also cryo-EM structures, which shows that the OSCA protein forms a dimer with two monomers sitting back to back. And there is one iron formation pathway within each monomer. However, when it goes to TMEM63 proteins, what they found is that these proteins only elicit tiny inward currents around 15 to 20 picoamps when overexpressed in EpIC cells. And also, no structure of these proteins is available. So this raises the question that whether the TMEM63 proteins really form mechanosensitive ion channels. And this is where we jump in. So we first just repeated the patch-cam recordings of TMEM63 mechanically activated currents in EpIC cells in this cell-attached configuration by applying negative pressures within the recording peptide to induce membrane stretch to activate the channel. And here, this is typically what the membrane looks like in the absence of the force. And here, this is the membrane with a pressure of minus 70 millimeter mercury applied. So when we just recorded the membrane stretch activated currents of human TMEM63B, but at different voltages from minus 100 all the way to the positive 100 millivolts. But we apply minus 70 millimeter mercury pressure. And you can say that we confirm that this very tiny inward currents at negative voltages, but we also say this very large outward currents when we go to the positive voltages. And this gives rise to profound outward rectified IV curve. And showing here, this is current traces with the membrane potential holding at a positive 80 millivolts and with increasing pressures within the recording peptide. And then we can get this beautiful sigmoidal mechanical activation curve. And more importantly, we found that just 1 point mutation in 63B can significantly change this deactivation kinetics. And this is when the currents, so the deactivation kinetics characterize how fast the current will decay after the pressure is removed. And because this is intrinsic property for the channel, we think that this point mutation data provide additional evidence clearly show that the TMEM63 proteins form mechanosensitive ion channels. So now with cryo-electron microscopy or cryo-EM, we determine atomic structures of TMEM63A and B. They are very similar. So I'm showing here TMEM63B. This is how this protein looks like. So you can say that different from the dimeric Oscar channel, these proteins are monomeric. And they share a very similar protein folding with one subunit of the Oscar channel. And this unique monomeric state of TMEM63 proteins was identified, was confirmed by extensive biochemical data and also by the subsequent cryo-EM structures of the TMEM63 protein from many other groups. So I think it's pretty clear that this protein form monomeric channel, but we still don't quite understand whether this has any functional or physiological relevance. Anyway, but when we look into the structure, we say that this protein is composed of two major parts. The first part is this transmembrane module, which contains 11 transmembrane helices from TM0 to TM10. And the second part is this large intracellular loop called L2 between TM2 and TM3. And the iron formation pathway is predicted to be formed by TM3 to 7. And you can say that the pore has a pretty much restricted region around here. So we think that this structure represents a closed state of the channel. So next we plan to solve an open state structure of the channel so that we can understand the activation mechanism. But this is very challenging because it's really difficult to apply physical force to a protein during structural determination with cryo-EM. So then we switched to disease-causing mutations in TMEM63B. We reason that some of these mutations may knock the channel in an activated state, so that which means we probably don't need to apply force, and then we are able to solve open state structure. So we focus on this V44M mutation, and this is the most prevalent mutation in TMEM63B. So this mutation is localized in the first transmembrane segment. The V44 residue points to the poor-landing TM4 and TM6, as you can see here. And this residue is also highly conserved in the TMEM63A, which is VP3, and the mutation is also here, VP3N. So we first demonstrated that this V44M mutation in 63B is indeed a gain-of-function mutation with different assays. So here are just one piece of the data. We overexpressed, when overexpressed in these heterologous HEK cells, either this V44M mutant of 63B or the VP3M mutant of 63A, they both induce significant cytotoxicity, as you can see here, compared to the wild-type protein. And here, this red, those represent a one dead cell, which is labeled by the cytotox, which is a DNA binding region that can only enter into the dead cells, but not the viable cells. Here, this is the quantification showing the percentage of the dead cells in the total cells. But then when we measured the ion channel activity, we found that the V44M mutation actually reduced the mechanically activated currents, as you can see here. And also, the mechanical activation curve is not much affected by this mutation. And this is also true for the TMEM63A protein. As you can see here, this is reduced mechanically activated current amplitude by this mutation. Also, the mechanical activation curve is not affected by the VP3M mutation. And in addition, we also found that this V2M mutation does not induce leak currents in both 63B or 63A, as shown here by these whole cell patch count recordings. And here is a positive control showing leak currents in a gain-of-function mutants of TRPM8. So this electrophysiological data does not explain the gain-of-function effects of the V2M mutation, as I showed you in the last slide. And then actually, this TMEM63 family is evolutionarily related to TMC family and TMEM16 family. And this TMEM16 family is calcium activated both ion channel and lipid scramblase. The lipid scramblase is a type of membrane protein or a transporter that allow bidirectional movement of lipids across the two layers of the membrane. And some of the lipids, like the phosphatidylserine or PS, they are typically only localized in the inner layer of the surface membrane, but they can go across the membrane to the outer layer through a lipid scramblase. And this externalized PS lipids can act as a signal to induce different cellular behavior, including cell death. And we can use alexin-5, which is a protein specifically binds to PS lipids, to characterize the lipid scramblase activity. So now we indeed observed that either the V44M mutant of 63B or the V53M mutant of 63A exhibit constitutive lipid scramblase activity, but not in the wild-type protein. Here, this is the quantification of the percentage of the PS-exposed cells within the GFP-positive cells. So this data potentially explain the gain-of-function effects of the V2M mutation. But the question is that how an ion channel is converted into a scramblase to transport lipids. So then we resolved with cryo-EM, we resolved the high-resolution structures of the TMEM63A V53M mutant, but not the 63B mutant. We don't know why, even we spent lots of efforts. Anyway, so within this TMEM63A mutant, we see two distinct conformations. And the orange one showing here represents the closed state. It's highly similar to the wild type 63A protein structure. But this green one here is different, and we sign as lipid open state because we say it binds extensive lipids, as you can see here, especially in this lateral cleft. And these cleft lipids go across the whole membrane in this conformation, which we think potentially provide a pathway for the lipid translocation. So here is another comparison between this closed state and lipid open states. And you can see clearly that in this closed state, the pore is apparently closed around this gate region formed by TM4, 6, and 0. But in the open states, this gate region is largely dilated to open a pathway for the lipids translocation. And this is further supported by the molecular dynamics simulation, showing how a molecule can move from one layer of the membrane to another layer through this pathway. And here is an animation to show this process. So the head group of the lipids is hydrophilic and will enter into the pathway during the translocation. And the tail of the lipids is hydrophobic and will also remain within the membrane. Okay, so now when we compare the structures between the closed and lipid open states, we found that the pore opening is mainly mediated by the outward movement of TM3A, 4A, and 5A, where other transmembrane helices remains unchanged. Here, this is showing here, this is in the side view, and this is in the top view, and the pore is around here. So I'm going to show you another animation demonstrating how the pore is opened by the movement of TM3A, 4A, and 5A. So this is in the side view. And now this is in the top view, and the pore is around here. Oh, sorry about that. The pore is around here. Okay, so you can see that the pore is directly opened by the movement of TM4A away from TM0 and TM6. So now the question is how the V2M mutation can induce such movement. And then we're looking into the TMM63A wild type protein structure, and we found that the V53 residue, which is here, this residue is part of a large network of hydrophobic residues as shown here in TM4A, TM6, and TM0. So we think that this hydrophobic network, which we call hydrophobic latch, will form hydrophobic interactions within the membrane to knock the TM4A against the TM6 and 0 in a closed state. And the V2M mutation would disrupt this hydrophobic latch and release TM4A to open the pore. And also, this hydrophobic latch is highly conserved in the 6,3B, as you can see here. So we think that this could be the mechanism how the V44M mutation could induce disease in humans. And another interesting thing I want to highlight is that this hydrophobic latch, you can see, is located just beneath the surface of the membrane. You can also see here clearly with the representation of the surface hydrophobicity of the protein. So here, this region represents hydrophobic latch. You can imagine that when the force applied to the membrane, and that will induce membrane stretch and also membrane thinning. And this will expose this hydrophobic latch to the extracellular hydrophilic environment and cause hydrophobic mismatch. And this is energetically unfavorable. So this would cause the protein movement to re-embed the exposed hydrophobic latch to re-embed within the thinner membrane. And this is what we observed in the open state of the TMEM63A mutant. As you can see here now, this hydrophobic latch has been re-embedded within the cell membrane again. So here you will see during the membrane thinning, how the protein movement will re-embed is a hydrophobic latch here within the cell membrane. Okay. So based on this, we propose a hypothetical model of how TMEM63 could be activated by physical force and also pathological mutations. So we think that in the resting state, this hydrophobic latch lock the channel in the closed state. And then when the force is applied to the membrane, that will cause membrane stretch and membrane thinning, and this will expose its hydrophobic latch. And this will cause the conformational change in the protein to open the pore. So this means that the protein, the TMEM63 protein, does not directly sense force, but sense membrane thickness. And some of the disease mutation may directly disrupt this hydrophobic latch to open the pore in the absence of the force. So that's all we learned from the TMEM63B gain of function V449 mutation. But what about the TMEM63A disease mutations? So showing here are the 5 point mutations initially identified in TMEM63A that cause hypomyelinating leukodystrophy, which is characterized by defects in the myelination of neurons in the brain. And functional studies have shown that all these 5 point mutations cause loss of channel activity in the TMEM63A. So it was generally, so over the past few years, it was generally accepted that the disease is caused by the loss of function in 63A. And because of the gain of function mutation in 63B that we just published, now we want to re-examine these point mutations. So we focus on this G567S mutation, and this one is localized in the middle of the pore TM6 around here. So what we first did is we just overexpress this G2S mutant and the wild type protein in the EpIC cells, and it's waited for 2 days. And then this is what we observed. So the G2S mutant induced significant cell death compared to the wild type protein. And showing here, this is a negative control with a Merck transfection, and this is a positive control with the gain of function TRPM8 mutant. And then we did a more quantitative assay with the cytox staining. Here is the bar graph showing increased cell death caused by the G2S mutant expression. Even though we found that this G2S mutant has slightly lower surface expression compared to the wild type protein, as you can see here. And then we also overexpressed this G2S mutant in the adipose follicle oversize by mRNA injection. And we just count the dead oversize each day. So showing here, this is the 4th day after mRNA injection. And here is the quantification showing much increased percentage of the dead oversize by injection mRNA of the G2S mutant. And here this TRPM2 gain of function mutant was used as a positive control. And again, we found that this G2S mutant has a little bit lower surface expression in the adipose oversize compared to the wild type protein. So because the G2S mutant has been shown to have lost mechanically induced activation, now the question is that how it can cause cell death here. So now we try to measure the constitutive channel activity with whole cell patchcom recording in EpIC cells and also 2 electron voltage clamp in the adipose oversize. But in both cases, we don't see any detectable constitutive currents. Again, here the TRPM8 and the TRPP2 channels were used as positive controls. And then we also measured the mechanically activated currents in EpIC cells, and we found that the G2S completely lost the activation. And this is consistent with the previous publication. And then, like what we did in the TMM63B, we performed nucleus accumbens assay with alexin-5 staining. And now we indeed say that we observe the constitutive nucleus accumbens activity in this G2S mutant, but not in the wild type protein. And here is the quantification. And then we also confirmed this in the xylophosph oversize. As you can see here, this alexin-5 staining in the oversize with expressing of the G2S mutant. And here, this is the quantification of alexin-5 staining is red fluorescence intensity normalized by the GLP fluorescence within the membrane. So now we also solved high resolution structures of the TMEM63A G2S mutant. And again, we see these two conformations, a closed state and a lipid open states. And in these lipid open states, we also see this dilated pathway for lipid translocation. And this pathway is highly similar to what we observed in the previous V2M mutant. So in both TMM63B and A, we found that the disease mutations they induce a gain of function effects by evoking constitutive lipid complex activity through opening a pathway here for lipid translocation formed by TM4, TM6, and TM0. And we think that this may represent a unifying mechanism for the human genetic disease associated with TMM63 genes. And we think that the future therapeutic development should focus on the poor blockers that inhibit this constitutive lipid complex activity. On the other hand, some really recent publication from other groups reported that TMEM63 wild-type proteins, at least when overexpressed in heterologous cells, can also function as lipid complex activated by force or membrane curvature. So in the future, we want to confirm whether TMEM63 proteins can form a novel family of force-activated ion channel and lipid complex. And if yes, how do ions and lipids could pass through a single TMEM63 protein? And what are the physiological roles of the TMEM63 mediated ion flux and lipid crumbling in vivo? And then lastly, how the constitutive lipid complex activities induced by the TMEM63 genetic mutations can cause the disease phenotypes. So with that, I want to thank my postdoc mentor, Jeffrey Hart, in Boston Children's Hospital. He actually gives me lots of freedom to explore different science. That's why I switched the lab focus from TMC1 to the TMEM63 proteins. I also want to thank all our collaborators for their very generous contribution to our TMEM63 projects. And also, I want to thank all my colleagues here at UW Medicine for their tremendous help in helping me set up my new labs. And of course, all my lab members. It is their hard working that allow me to present here. And definitely need funding agencies. And last, I want to emphasize that the cryo-EM data is collected in the Harvard Medical School Cryo-EM Center and also UW Medicine Cryo-EM Research Center. And so that's it. Now I'm happy to answer any questions.
Thank you so much, Wang, for this very clear talk. We already have quite a few questions in the chat, so I'll start with that. Damien asked a question, but then told us not to. You answered his question during the talk, so we can skip that. So Esteban Suarez Delgado asks, thank you for sharing your beautiful work. I had a naive technical question in your experiments. Adding negative pressure sometimes deforms the membrane in the patch inside the tip. Could that affect how the channel responds? If so, how do you do the quality control of your patches? Have you measured the response with positive pressures?
Yeah, that's definitely a good question. We actually do not calibrate the membrane tension in our pipette. Um, so that's correct. So sometimes we also say that there's some like a spontaneous membrane curvature when we form a seal, even before we apply pressures. But we normally only measure the currents when we apply pressure, but we haven't correlated this pressure to the membrane curvature. So some labs is doing that. I should say the best correlation should be correlate the membrane activation and the membrane curvature. But measuring membrane curvature is very difficult. So we normally only uh, measure the pressure and then plot the currents against the pressure. So we haven't done any calibration to the membrane curvature in our experiment.
Yep. Okay, so the next question is from Lucy Forrest regarding MD simulations. Were the MD simulations of TMEM636A B53 mutant carried out with a standard thickness membrane patch or a thinner one that matches the hydrophobic thickness of the mutant? Did you see reproducible reversible lipid movements?
Yeah, so we haven't tried different membrane thickness in our simulation. So in the simulation, what we did is just like embed the protein within like a POPC, POPE. I think that's represent the native lipid, so it's in the normal membrane thickness. So that's something we are doing now. So we are collaborating with simulation lab that we're trying to use different lipids to mimic different membrane thickness, and we want to see the protein behavior in that system. But that's what we are doing now, actually.
Okay, thank you. So Jorge Granul asks, very nice work. Do you see an area expansion, so change in surface, between the closed and open state, as would be predicted from an ion channel that is activated by membrane tension? If so, how large it is? Is it?
Yeah, I don't quite catch the question.
So do you see the ion channel, so the protein, when it's closed and open, do you see between these two states, closed and apparent open state, do you see an enlargement in the surface area of the channel of the?
Oh, I see. Yeah. Actually, we do see some difference. I'm showing here. I think the best way is to go here. So first of all, we say that the membrane or the detergent, the micelle around the protein becomes thinner. You can see that the thickness in the closed state is about 50 angstrom, but in the open state, it's reduced to about 44 angstroms. And the other thing that we do say the surface was expanded at some level, because here you will say in the open state there's an outward movement or this kind of flower opening movement of TM3A, 4A, and 5A, and this indeed expanded the surface area a little bit. But we're not quite sure whether this has anything to do with the opening of the channel by the force. But this is just what we observed in the closed and open states by these pathogenic mutations. But I think in the future, and also what we are doing now is we are looking into how force activate these proteins. And what we're doing now is trying to use different lipids, so we can reconstitute our purified protein into lipids, and then we can control the membrane thickness by using different lipids with different lengths of the tail, right? So now we can say whether we're going to see open state when we use the membrane thinner membrane, or the closed state using the thicker membrane. And also those are something that we are doing now.
Okay. So the next question is about seeing currents in this positive voltages, right? And this is exactly what I was wondering. So what is the physiological relevance of mechanosensitive currents measured at plus 80 millivolts, right? So when would that physiologically come into play?
We don't know. Because first of all, so far we don't have much information what those protein is doing in vivo. And the second was you mentioned, yes, they showed large currents at positive voltages. And it seems like at the physiological conditions, this is maybe the protein may not experience its positive voltages. So we have no idea whether those positive voltage large currents will have any like, physiological relevance. But another direction in my lab is we're trying to look into the in vivo physiological function. So once we know more information about that, I think we're going to answer this question: how this membrane, how this protein channel activity, either at elective or positive voltage, that will contribute to their physiological function in vivo. But we don't have that information now.
I'm just going to pop in one question for me before I move on to the other, because we're already talking about this strong dependence on positive voltages. Is there anything in the structure that explains this?
No, from the structure, we have no idea about this. Yeah. I would say even in our structure, we also see some kind of rectification. But TM63 protein reactivity is profound. We have no idea. Probably there may be some like a voltage sensor domain in the protein that will allow it to modulate the back voltage. One project that we plan to do is to try to mutate all those charged residue in the protein and then try to say whether they eliminate this rectification.
So the next question is, how do you envision that understanding the molecular defects caused by naturally occurring congenital mutations can improve the life of those affected by them?
Can you say it again?
So how do you picture that understanding the molecular defects caused by naturally occurring congenital mutations can improve the lives of those affected by them? So how will us understanding the structure and function of this protein at atomic detail help people who actually carry those mutations?
Yeah, I think that's definitely a good question. So, I mean, in order to develop therapeuticals, we need to first know the basic mechanism, right? For example, in the TMM63A, I mean, the previous concept is that those mutations cause loss of function in human patients. So now you would say, okay, now you want to develop the treatment, you need to correct this mutation or use some like agonist, right, to correct their function. But now if we found that if this disease is caused by the gain of function, the lipid constitutive nucleus complex activity, now the strategy will be to develop blockers, right, to inhibit that lipid complex activity. So I think this structure function relationship study will give us the guidance to develop therapeuticals.
Yeah, that makes sense. So there was one question from Connor who had to leave early, but he popped it into the panel discussion. I wondered whether, in addition to your convincing scramblase data, where the V44M mutant might introduce a novel alternative translation initiation site. So if you delete amino acids 1 to 43, can you get any channel activity with some weird behavior?
Oh, yeah. Yeah, that's a good question. We have some data for the TMM63A that we haven't published. And we are going to publish, I think, pretty soon. It's like we can introduce some additional mutation and we can recover the ion channel activity. So, which means like the mutation, for example, in the TMM63A, this mutation, they cause both loss of function in the channel activity, but also gain of function in the lipid complex activity, right? But the protein, we should say, it still have the potential to be activated to function as ion channel activity. I didn't show data here, but the loss of function in the channel activity is because some very different mechanism, and we can correct that mechanism through additional mutation to the protein. Yeah, so I'm not sure whether I answered the question from caller. If not, I think we can talk about it later.
Yeah, he can let you know. Okay, there is another question in the chat. Does the rectification with voltage reflect voltage-dependent gating or single-channel conductance rectification?
Yeah, that's a good question. I think it's because of the voltage-dependent gating. Because we try to measure the single channel currents at the positive voltages, but we're still not able to detect any single channel currents. So first of all, the TM63 has extremely small single channel currents. Is even not detectable with the conventional single channel recordings. So some people use the noise analysis to estimate that the single channel conductance of TM63 proteins is it's less than 1 picosiemens. That's extremely small. So we think that if at the positive voltages, if this is because of the increased single channel currents, probably we can record that single channel currents. But we have tried many times trying to record the single channel currents at the positive voltage, but we're still not able to see any. So we think that most likely this dependence is because maybe the voltage can regulate the gating.
Thank you. So we have a few more questions in the chat, but Marcel has his hand up. Marcel, do you want to go?
Sure. Hi, Wang. Great talk. So I'm just curious, is there anything known about whether or not the scramblase and the ionic conduction are coupled in some way? So you have all these loss of functions. You said you could introduce this mutation that brings back the ionic conductance. Does it inhibit the scramblase, or are they completely separate?
Yeah, that's a very good question. So as I mentioned, this is a big question that if the protein function as both ion channel and scramblase, how the ions and lipids will coordinate, will pass through the single pore. Actually, what we found is like the ions and the lipids, they may share part of the pore, but not totally overlapped, which means can I say here? We think that the pore at the superficial aspects here, the lipids can go away, can go across the pathway, but they will not go deep into the pore. So within the deep of the pore, and that is the pathway for the ion channel. So which means they share the same region for the pathway, but they are not totally overlapped.
So do you think you can develop blockers of just the scramblase and not the conduction?
Yeah, so one of the strategies that if the pore blocker only binds to around here, let's say, but not the deep of the pore, and this may only block the lipid translocation, right? But if you have a small molecule that can bind to the deep of the pore, and then the lipid translocation may not be affected. So I think it's possible that you can develop different chemicals that can specifically inhibit the ion activity and also lipid translocation. And this is something that we're trying to develop actually using that as a tool to dissect what is the ion chain activity and the lipid scramblase activity, what is the physiological roles of these two molecular function in vivo. But so far, so if you do a lookout in mouse, you look out the both function, right? If you do some like gain of function, and then some of the mutations may also affect both molecular. So what we want to do is that we want to find either a chemical or a mutation that will only affect one molecular function, and we can specifically study the physiological roles of that function in vivo. Yeah, so there's still lots of questions here. And also we have some unpublished data I didn't show here, but you can pay attention to our next publication. We are going to show some of this data in our next publication problem is in this year.
Thank you. So we've got 2 more minutes for maybe 1 or 2 more questions. So Liam is asking, activation of TMM63 by hydrophobic mismatch after membrane thinning is an interesting mechanism. Do you have a hypothesis to explain how current decay occurs more rapidly for the mutants after pressure is removed?
After the mutants, sorry, I lost my mind. I didn't catch the question. Can you say that again?
So do you have a hypothesis to explain how current decay occurs more rapidly for the mutants after pressure is removed?
The current decay.
There was one on the slides, yeah.
I'm not quite sure about the current decay. I think the current decay is that when the force is removed, and then right.
It was in one of the slides. If you go through, one of the mutants had just a quicker decaying current. So I think it was this one. For example, even this one. Yeah. For example, the pink one has P492. It has a quicker current decay than the wild type. So the question is, do you have a hypothesis to explain what's causing this?
We don't know, but it's interesting. This protein mutation is localized in the TM4. So let me just quick show here. So that mutation is actually localized here in the middle of TM4. So we think that because the TM4 is involved in the pore opening and closing, right? So we think that protein mutation may just make the TM4 prone to be in the closed state. So once it opens, and it will quickly goes to the closed state. That possibly be the mechanism, but we have no solid evidence for that.
Okay. I think we are at the hour, but there's just one more question in the chat, so I think we can just have enough time to address it. So Damien Bell has been circling about this. So he has noted this very large current in on-cell experiments, right? So he's wondering how could you have such high on-cell currents with such a low conductance? Do you know that you have super high expression of these ion channels on the surface of the cells, or? Yeah,
that's right. So I think we have pretty much high expression in the surface. We overexpress our protein, and we can say when we patch the cells with very strong fluorescence and no fluorescence, and we can say we normally can see much larger currents in the very strong fluorescence. So I think the protein expression seems to be correlated with the current amplitude. So that's why I think we believe that this large current I'm not sure. So that's why we think that in this large current comes from probably as a lot of proteins on the surface in our patch. But I'm not excluding the possibility that at the positive voltages, the single channel currents may also increase. That may also contribute to these large currents here.
Okay. Thank you very much. I think we are officially at the end of the seminar. Wang, thank you so much for joining us and for sharing your great data. Thank you so much. Okay, until April then, everybody. Thank you.
Okay, thank you.
Bye-bye.